body. What happens after that, after we've completed building all the structures of the body, is really, really rapid growth, so that by 10 weeks we have a big increase in size. You can see fingernails, eyelashes. By 14 weeks, even larger, and by 18 weeks, the fetus is about 6.5 inches long, weighs about 3/4 of a pound, and it's moving, reacting to sound, to light, to touch, and to pain. So that's what happens, but it doesn't really answer the question of when life begins, because all of this information leaves us with what I call the continuum problem. And that is that because development proceeds continuously from the one-cell stage up to birth and beyond, many people set arbitrary points for when they think life begins or for when they feel humans have dignity and human rights. So a lot of people will point to that blastocyst stage, the point at which we have the first distinct cell types in the embryo, at about 4 days post-sperm egg fusion. Some people like implantation, or the point where the embryo first establishes a relationship to the mom. Many people point to an event I didn't discuss, known as primitive streak formation, which happens at about 14 days post-sperm egg fusion. And people like this point because it's the earliest stage at which we know where the nervous system is going to form in the embryo. But you can go later than that, kind of analogously to brain death. Many people point to brain life, or an ill-defined point at which we have the onset of mature patterns of brain activity, somewhere around 5 months post-sperm egg fusion. But you can go even later than that. Peter Singer at Princeton is famous for having proposed self-awareness as the point at which meaningful human life begins, an event that he claims happens at about 28 days after birth. And for myself as a developmental neuroscientist, my personal favorite is, of course, brain maturation, something that doesn't happen until you're at least 25. So I used to tell my teenagers, I can send you back to the human parts factory, you're not even human yet. But I think what's evident from this is that all of these are arbitrary criteria, and choosing among them is a matter of taste or utility or, more likely, power, not a matter of science or reason. So how then does science address this question? Well, in order to answer this question, we have to ask two related questions. The first one is, when is a new cell type produced from the interaction of sperm and egg? And the second one is, what's the difference between a human cell and a human being, or a human organism? So let's tackle that first one first. Thankfully, scientists use two very, very simple criteria to determine when a new cell is formed. Changes in cell type are detected either by changes in material composition, so what a cell is made out of, and this typically is changes in gene utilization, or by changes in cell behavior, and very often these two things come together. So if you alter what a cell is composed of, you can also alter the types of behaviors it's capable of showing. I can assure you these two criteria are unambiguously used throughout the entire, and uncontroversially used throughout the entire scientific enterprise to determine when a new cell type is formed, either in the laboratory or in an embryo. And of course, logic also requires that if a new cell type is formed from one or more pre-existing cells, those cells have to cease to exist before we can say we have a new cell type. So, based on these criteria, is the product of sperm egg fusion a new cell, or is it just a modified gamete? For example, you often hear the product of sperm egg fusion or fertilization referred to as a fertilized egg. So is a fertilized egg a meaningful term? Is it a scientific term? Well, I hope to convince you that the answer to that is no. A fertilized egg is not a meaningful term, but rather that a new cell forms at the instant of sperm egg fusion. So this is a scanning electron micrograph of a larger egg cell and a sperm cell fusing to it. And upon sperm egg binding, the plasma membranes, or the outer surfaces of these two cells, will fuse together very, very rapidly in a well-studied molecular event that takes less than 1/4 of a second to complete. So there is an instant of fertilization, it's about 250 milliseconds, where two cells go to one cell. And when that happens, all of the lipid and protein that used to be present on the surface of the sperm now incorporates into the surface of this new cell, changing its molecular composition. In this fusion of the outer surfaces of these two cells, the sperm is rupturing and it's ejecting all of its cellular contents into the inside of this new cell. So the sperm clearly as a separate cell has ceased to exist in this instant of sperm-egg fusion. What about the egg cell? So this is a one-celled embryo shortly after sperm-egg fusion. And following the fusion and destruction of the sperm, the resulting cell has a new composition that's distinct from that of an egg. So we've already talked about the incorporation of lipid and protein from the sperm into the surface of this new cell. But the changes go further than that. This cell also has a new genetic composition with half of its DNA from mom and half of it from dad. But moreover, this new cell also radically changes its behavior and acts in a very un-egg-like manner. So the entire raison d'etre of being an egg cell or gamete is to find a gamete of the opposite type and to fuse with it. And in fact, a fully mature egg, if it doesn't fuse to a sperm, has a very short lifespan. It only lasts about 24 hours. But within 1 to 3 seconds of the fusion of sperm and egg surface membranes, this cell will initiate a molecular cascade that directly antagonizes the function of a gamete, that acts exactly in the opposite interest of an egg cell. So it initiates a molecular cascade that will result in the deposition of enzymes into the space around the cell between that protein coat, the zona pellucida, and the surface. And the function of those enzymes is to destroy all the sperm binding sites. So the entire primary function of an egg is to bind to a sperm, but the very first action of this new cell is to prevent binding of a sperm. So the egg cell clearly also as a separate cell, as a haploid gamete designed to fuse to a sperm, has ceased to exist. And a new cell with unique material composition and unique behaviors formed in this instant of sperm-egg fusion. So what is this new cell? Well, its scientific name is a zygote, or one-celled embryo. But the challenge is, looking at this picture is not terribly informative. So is a zygote a new individual human? Or perhaps some other kind of human cell? A pre-embryo, maybe, something that's a cell that's on its way to becoming a human being but isn't a human being yet. And the challenge we face is that at early stages, recognizable human form and function are not evident. I don't look at those pictures and go, oh, it's a baby. And I'm a scientist. I look at these pictures all the time. We don't have an emotional response to these things. They're not part of our daily intuitive human experience. So how do we know that a zygote or a morula or a blastocyst is a human being and not merely a human cell or a ball of human cells? Well, to answer that question, we have to first answer, how do cells behave? So this is a picture of a typical cell in culture. And most cells in your body, if put into a laboratory culture dish, do nothing. They'll survive for some period of time and eventually die. Some cells will divide, but when they divide, they're just like Xerox machines. They make identical copies of themselves. And that's all they ever do. Rare cell types, for example, stem cells, can generate progeny that have different properties in different culture environments. So, for example, here, if you culture a stem cell in either low levels of oxygen or high levels of oxygen, the cells here are stained just to reveal the molecules that are present on their surface. So different colors reflect different types of cells. What you can see is that in low oxygen, all the cells look pretty similar, but in high oxygen, you can make cells with different colors, different properties. But in all cases, what you get is a tumor. You get production of cells in a random pile with no relationship to each other and with no coordinated function. So all cells can do is produce either copies of themselves or tumors. In contrast, how do embryos behave? Well, in an appropriately supportive environment, the embryo undergoes a self-directed, organized pattern of development to generate the fetus. It doesn't function as a single cell or even as a group of cells, but rather as an organism. So, what's an organism? Merriam-Webster gives us a couple of definitions. One, a complex structure of interdependent and subordinate elements whose relations and properties are largely determined by their function in the whole. Or 2, an individual, constituted to carry on the activities of life by means of organs separate in function but mutually dependent, a living being. Now, both of these definitions stress exactly the defining feature of an organism. So an organism is a complex thing that's made up of parts. And those parts do different things, but all those things work together. And they work together for the good of the thing as a whole. I would also note that people will claim the term human being is not a scientific term, and yet a living being, part of the definition of an organism, of the genus Homo sapiens would be a living human being. So human being is simply a synonym for human organism. This is a paper published relatively recently in 2016 that lists a long and complicated list of characteristics of organisms to try to distinguish them from non-organisms. But I think they can be simplified into 4 basic traits. Autonomy, and I would add to this list the capacity for wound healing. So if there's damage to an organism, the organism autonomously repairs back to its native state. Integration, development, and adaptation, or AIDA if you like opera. So the features of organisms, in my more simplified list, are development, so growth towards a characteristic, mature state that functions as an integrated whole, what philosophers would call the perfection of form. Autonomy, self-directed repair of injuries such that the unified function of the entity is preserved. Adaptation, healthy adjustment to changing environmental circumstances. And integration, the coordinated function of parts to support the function of the entity as a whole. Now, I would also note that these are discrete, observable, scientific characteristics. They're not mystical qualities, and they're not matters of opinion, and they're not debatable. Either they exist or they don't. And when they do exist, they unambiguously distinguish cells from multicellular organisms. So when does organismal function begin? I think most of us would agree this is an organism. It's a complex entity comprised of parts that work together for the good of the entity as itself, as a whole. And I think even many of our opponents would agree that this is an organism. It's got all the same structures that first one did. But is this an organism? I have a friend who's an attorney, and every time he sees this picture, he goes, "That's not an organism, that's a bear aspirin." And I have to say, it does kind of look a little bit like a bear aspirin. But I hope to convince you that from the one-cell stage forward, embryos are unambiguously organisms. And I say that not because I like that conclusion, but because it's the only conclusion that's consistent with the facts. Let's go through those characteristics we just listed. Growth towards a characteristic mature state. Well, this is the whole business of embryonic development, is to proceed directly within the first 3 seconds towards maturation of mature human form. Autonomy, repair of injuries such that the unified function of the organism is preserved. Well, embryos are actually really good at repairing injury, and I'll give you just one example, that of pre-implantation genetic diagnosis. So what you're seeing here is an embryo immobilized here against a smooth glass pipette with a sharper pipette going in to remove a cell from that embryo. So typically a single cell is removed from an 8-cell embryo, and those biopsied embryos are maintained in a laboratory dish while a genetic analysis is performed on that removed cell. And if the embryo passes the test and it's genetically normal, then it's transferred to the uterus of the intended mom. And remarkably, embryos recover from what could only be seen as a completely catastrophic injury. I mean, to remove an eighth of your body at such a tender age is pretty impressive. But they regenerate those missing parts, such that there have been thousands of live births from biopsied embryos worldwide. What about adaptation? Well, again, embryos are good at this. I'll give you one example only, that of ectopic pregnancy. So in most cases, when an embryo implants someplace outside of the uterus, it dies because it's usually because it's unable to establish an adequate placental circulation with the mom. So you need to have a placental circulation very early, or you just don't get adequate oxygen and nutrition. But in rare cases, if an embryo is fortunate enough to implant in a favorable location, you can have cases like this. Back in 2003, this little baby here, Nahalanisi of Filipe, South Africa, went all the way to term, 9 months, inside her mother's liver. So how could that happen? Well, it happened like this. She took a wrong turn in the fallopian tube, and she ended out in the abdominal cavity of the mom. And the liver is a great organ to implant on because it's very soft, and it has tremendous amount of vascularization. So she was able to establish a placental circulation with her mom through the liver. And at the end of 9 months, I can't even say a C-section. They cut open the mom's abdomen, made a little slit in the liver, and took the baby out. And look at them, they're both perfectly healthy. Now, if a baby can go 9 months normally to term inside her mother's liver, that's, I think, great testimony to the adaptive abilities of embryos. Lastly, integration, or the coordinated function of parts to serve the entity as a whole. Again, embryos do this very, very well. I'll give you just one example. These cartoons show the initial stages of cell division from a one-celled embryo to a two, to a four-celled embryo, and to the blastocyst stage. And an enormous amount of work has demonstrated that the pattern of cell division at the two-cell stage determines what cells will contribute to, what structures they're going to contribute to later in development. So if you see a pattern that looks like this, then the pink cells and the red cells are going to contribute preferentially, albeit not exclusively, to the postnatal body. And the blue cells and the green cells are going to contribute preferentially, although not exclusively, to the placenta. So this could happen in a lot of ways, but we know it happens because the cells at the four-cell stage have different developmental properties. And we know that from an experiment like this. So if you take 3 embryos that have the same pattern of development, so you can recognize the red cells and blue cells and green cells since they're not actually colored in real embryos, and you take 1 cell of each type from each of those 3 embryos, aggregate them together so you get 3 red cells, 3 blue, or 3 green, transfer them to a surrogate mom. The red cells can make a live-born pup in about 85% of the cases, whereas the green cells never do. And what that tells us is this, that at the 4-cell stage, cells have different developmental capabilities, different powers, and that in normal development, they need to work together in order to make a complete pattern of development. So I think in 2014, I published a very long review in the Journal of Law and Public Policy, which you can find by searching my name and When Does Life Begin, that reviews an enormous number of events that are happening at fusion within the first 3 minutes and up through the first few days of life that make it absolutely clear that sperm egg fusion sets off a molecular cascade that will control events that take place weeks or even months or even into the maturity of this animal. None of these events are required for the cells to remain alive. They only make sense as part of a developmental sequence directed towards producing more mature stages of life. And that can only be produced by an organism. And while some scientists may dislike this or deny this conclusion, it's supported by hundreds of peer-reviewed papers and is absolutely uncontested in the literature. It's not that I've selected the papers I like that prove my point. There are no other papers. Embryos are unambiguously organisms. So let's conclude from the facts. A new cell, the zygote, with unique material composition, genes, and behavior comes into existence at the instant of sperm egg fusion. It exhibits all of the characteristics of a full and complete, albeit immature, human being. Therefore, human life commences with the onset of organismal function at sperm egg fusion. So human zygote is a human being. The conflating of the question of when life begins with the question of when life has value. So what do we think about human dignity? I'd like to refer you to a really nice analysis by Frederick White in 2013 in the Lineker Quarterly, where he goes through, I think, in a very comprehensive way, all of the kinds of ways you can think about dignity. And he argues that there are they all break down to 2 essential views. Either that personhood is a state of being inherent and essential to the human species, or 2, that personhood is a conditional state of value defined by society. And White concludes, personhood is an essential characteristic of the human species and is not a conditional state dependent upon circumstance, perception, cognition, or societal dictum. Now, this topic of dignity could and indeed has been the topic of entire semester-long courses. I really can't dwell on it, but I will note that White's conclusion is my conclusion, and it's also in very good agreement with the teaching of the Catholic Church, where in the Catechism at 2258, it states human life is sacred. From its beginning until its end, no one can, under any circumstance, claim for himself the right to destroy an innocent human being. This is a very strong prohibition, even in the context of the Catechism. It doesn't say you don't have a right to kill people. It says you don't have the right to even claim for yourself or for anyone else, you pro-choice Catholics out there, the right to destroy. Can't claim the right, independent of actually doing it. So human life and human dignity, none of us were a sperm and an egg. I think as Janet said earlier, those are human cells. They're living, but they're not human beings. And yet, once they combine to create a human organism, it is a human being, an immature human being that grows up to a mature human being. And therefore, embryo rights are human rights. So in the last few minutes, I'm going to talk very briefly about some medical and research technologies that need to be informed by this understanding of when life begins, and some of its uncomfortable implications. Many forms of contraception are not just bad for all the reasons we've heard already today, but they're bad because they're embryocidal, and they kill a developing human being. The Centers for Disease Control, in its most recent review of contraception, indicates that there are 61 million women of reproductive age, and their use of contraception breaks down in this way. So there's a lot of information in this slide, but one of the things I'd like to point you to is that 38% of women use no contraception, and 62% use some form. The way this graph depicts it, it's hard to break it down, but if you look at the tables, it translates to about 30% of American women using hormonal contraception or IUDs, which is about 18 million women. A recent review that appears in Lineager Quarterly by Donna Harrison demonstrates, I think very convincingly, that hormonal contraception and IUDs are likely to work both by preventing ovulation, but also by killing embryos. And given that 18 million women are using potentially embryocidal contraception, if only 10% of the effective contraception is embryocidal, if only 10% is actually killing embryos as opposed to preventing sperm-egg fusion, up to about 1 million embryos a month are lost due to the use of contraception. I think this is a feature that very few people appreciate about contraception, because this puts contraception on rank with direct abortion for the number of human lives that are lost. Let's move on to a topic that nobody has mentioned yet today, but I think is very much related to the theology of the body and the instrumentalization of sex, which is assisted reproductive technology. So ART is widely used. Again, this is a report from the CDC, which indicated in 2015 there were over 182,000 ART procedures in the United States, resulting in about 60,000 live births, or 1.7% of all infants born in the United States. An important question to ask about this is how many embryos are destroyed in the process of performing all of these procedures? The answer to this simply is that we don't know in the United States, but that in 2012, in the UK, a report to Parliament indicated that over a 10-year period, roughly between 91 and 20-year period, between 91 and 2012, 3.5 million embryos were produced. There were 1.4 million transfers and about 250,000 pregnancies, with 840,000 embryos frozen, 23,000 of which were destroyed at some point after thawing, but 1.7 million embryos were discarded, all in the course of pursuing that one magical special baby. ART also constitutes a medical risk for women, and I'll give you just one example. This was a study done in the Netherlands that showed there were 48.5 deaths, maternal deaths per 100,000 due to IVF or IVF pregnancies, compared to only 12 deaths for spontaneous conception, a fourfold increase in maternal risk, something that's not disclosed to women when they elect to do this procedure. Lastly, ART constitutes a medical risk for children conceived in vitro. So one of the negative consequences of abandoning a correct theology of the body is this one. We pass it on to our children, and I'll give you just a couple of studies. Published in 2017, this study looked at over 5 million infants, and it shows statistically significant increases in low birth weight, preterm delivery, cardiac abnormalities, central nervous system abnormalities associated with cognitive impairment, urogenital system abnormalities, and musculoskeletal disorders. Similarly, this paper, published the same year, looking at 300,000 births, statistically significant increases in stillbirth, low birth weight, very low birth weight, very preterm birth, and neonatal death, and statistically significant increases in major birth defects, including cardiac, urogenital, musculoskeletal defects that are doubled after intracytoplasmic sperm injection, which accounts for about 70% of all ART procedures worldwide. And this is only talking about the babies. We have follow-up data from a lot of these children now that are young adults, since we've been doing ART for decades. And there are very troubling increases in a number of rare medical conditions, very serious medical conditions in this population that are not seen in naturally conceived individuals. Lastly, many research technologies destroy human beings. So it's pretty obvious that fetal tissue research incentivizes the destruction of human beings for science and therefore cannot be seen as licit. Similarly, human embryonic stem cell research and therapeutic cloning for stem cells also destroy embryonic human beings to obtain their stem cells. We haven't actually accomplished human cloning to adulthood, though we've done it in animals, and in those studies, typically 150 individuals die for every live-born animal we produce, and all of the cloned animals are epigenetically abnormal. So even if we succeed in cloning humans to live birth, this would be an intrinsically very immoral procedure. So medical and scientific technologies, many forms of contraception are embryocidal. Assisted reproductive technologies destroy enormous numbers of living human beings. ART is dangerous both for women and for the children conceived by this means. And fetal tissue research, embryonic stem cell research, therapeutic cloning all rely on the destruction of human beings. All right, practical suggestions, and this will only take maybe a minute. You cannot leave ethics to scientists. Scientists as human beings are no more inclined to unethical behavior than anyone else, but a culture of science strongly incentivizes a strictly utilitarian view of biomedical research. And scientists overwhelmed by the explosion of information simply have no time to consider ethical judgments. This is a graph of the number of papers added to the world's largest database of scientific publications, PubMed. This is when the first textbook in my field was published in 1970, and this is when I was studying for my qualifying exams. And as a graduate student, I really was quite diligent, and I decided I was going to read the entire field of developmental neuroscience. 385 papers. I made a stack about this big in my apartment. Currently, over 300 papers a month show up in my field. It's impossible. It's impossible for someone to stay on top of a scientific area and have time for ethics. Moreover, there can be no simple view of this. Contraception and ART are widely practiced, and yet many people are very unaware. And they're focusing in that little Venn diagram I showed you at the beginning, on the benefits to humans within that little small window of focus. Scientific technologies that destroy embryos nonetheless have potential to increase knowledge and relieve human suffering. So to make a cogent case for restricting or prohibiting a particular technology, we need to acknowledge the potential benefits. We need to see things from the perspective of the people who are the advocates, while still advancing a clear argument for why the technology must be limited. So, get educated. I commend you. That's what you're doing here. Know the facts and be able to argue them so that we can take personal responsibility for what happens. Get involved. There are many opportunities for community members to get involved in regulating scientific practice in hospitals and in universities. Get on the ethics board. Have your voice heard. And be proactive. Beginning in high school, curricula need to be implemented that discuss how ethics shapes a just, moral, and effective biomedical research and reproductive practice. My most important conclusions: scientists unambiguously indicates human life begins at sperm egg fusion, and we as citizens and as Christians must take personal responsibility for the just treatment of human lives at all stages. Thank you.